I'd now like to introduce Dr. Antti Iivanainen. After receiving his DVM degree from the College of Veterinary Medicine in Helsinki, Finland, Dr. Iivanainen worked as a district veterinarian in northern Finland. He then turned to academia and received research training at the University of Oulu and at the Karolinska Institute in Stockholm, Sweden. Since then, he has worked at the University of Helsinki, where he is currently leading the bachelor's program in veterinary medicine. His research group is interested in interactions in development. As always, please post any questions you have for Dr. Iivanainen in the chat box, and he will answer questions from the audience as part of the live Q&A after his presentation. Welcome, Dr. Antti Iivanainen.
Good day. My name is Antti Iivanainen, and I come from the University of Helsinki in northern Finland, northern Europe. And the presentation of today is on the studies on canine hip dysplasia. The outline of the presentation is here. We will start with the normal development and anatomy of the hip joint, and then proceed on to discussing hip dysplasia as a developmental disorder. We then briefly discuss some of the risk factors that have been suggested to promote to the development of the disease. And then some genetic background studies. Then I will describe our own studies. There are two sets of studies. One set of studies was conducted on German Shepherds, and the other set of studies was conducted on 10 breed large cohort of dogs. Then I have some summary and conclusions, and a short glimpse of the future, how I see it. The pelvis and femur are ossified during the fetal life, so that ossification begins in the ileum around 40 days of gestation. This is followed by the appearance of an ossification center in the ischium at about 45 days, and finally in the pubic bone just before birth, 55 to 60 days of gestation. These figures are from Beagle. The acetabular bone, which is in the center of acetabulum, ossifies only after birth. The femoral shaft is ossified during the fetal life, but the ends of the femur are not ossified at birth. All of this means basically that the hip joint in the young animal is very prone to mechanical forces, and this is of course quite natural as the animal is growing, and the joint has to accommodate to the growth of the animal. In the mature pelvis, we can see in this picture, which is a 3D surface rendering of computer tomography image or stack of images. We can see here on the left, we can see a view from the caudo-dorsolateral point of view. The wing of ilium is seen clearly, and to the caudal there is the ischium. In the figure, we have marked the edge of the acetabulum, the dorsal edge, and the cranial lateral edge, which are something that are evaluated in radiographic images when screening for hip dysplasia. In addition, not marked in this figure, there are some osteophyte formations indicative of osteoarthritis, which typically form in the neck of the femoral head. On the image on the right, this is ventrodorsal view. One can see how the Norberg angle is defined. So it is defined by the craniolateral acetabular edge and the center of the femoral head. This figure illustrates the normal structure of articular cartilage. It is divided in three zones. The superficial zone is on the top, marked A, and this contains quite dense population of chondrocytes, which are more or less parallel to the surface of the joint. The superficial zone is secreting lubricin, which is a lubricant and makes it easier for the joint to function. The middle zone, marked as D, contains more round and a bit larger chondrocytes, and is very active in producing the matrix, which is predominantly containing different proteoglycans and type 2 collagen. The deep zone contains stacks of chondrocytes which are more or less perpendicular to the surface of the joint. And between the deep zone and then the ossified subchondral bone, one finds a mark which is called a tight mark that separates the bone from the cartilage. It may be worth emphasizing that the matrix in cartilage contains a lot of water. The proteoglycans contain molecular groups of the structure like chondroitin sulfate and keratan sulfate, which are very hydrophilic and attract water. And this is quite important for the physical properties of the tissue. The chondrocytes take about 2% of the volume in the cartilage, which is devoid of vessels normally and devoid of nerves. The chondrocytes are more or less arrested in a state which is called the pre-hypertrophic state, and they are undergoing a minimal turnover. Their main function is to maintain the integrity of the tissue by producing the matrix material. Now we can go to the disease, and it is considered a developmental disorder, but non-congenital. So the animals are basically born without any signs of illness, but the earliest signs can be detected, depending on the case, already at 2 weeks of age. The disease normally manifests itself in rapidly growing young individuals, and most often the large-sized breeds are affected, but the disease is not confined to these breeds. German Shepherds, which is an important breed in our studies, displays prevalence between 19 to 45% in the studies that I have seen. But according to the statistics at the Orthopedic Foundation for Animals in the US, the frequency as measured by dysplastic evaluations out of all evaluations of the hip joint varies from 0 to 71% between different breeds. The main clinical signs in the disease include joint laxity and varying degrees of pain. They are accompanied by morphological signs which can be quite variable and not necessarily correlate with the clinical signs. The disease is considered chronic and progressive, and very often progresses towards osteoarthritis, which can be a very painful condition and severely impact on the quality of life of the animal. In this figure, we can see a typical radiograph of the hip when they are evaluated for hip dysplasia. On the right, we see a normal radiograph where one can observe that the fit of the femoral head in the acetabulum is quite good, and the femoral head sits in the acetabulum quite snugly. On the image on the left, on the other hand, there are dysplastic changes, and the femoral head does not fit well in the acetabulum. The joint articular surfaces are not congruent, and one can also observe some osteophytes in the femoral neck. So about the phenotype of hip dysplasia. The most common way to diagnose hip dysplasia, and at least in Finland, that is the predominant sort of official way, is through radiological imaging. And the radiographs are taken in what we call a hip extended position that you saw on the previous slide from the ventrodorsal projection. And from these radiographs, different subtraits are assessed, and an aggregation or an aggregate score of the substrates is somehow combined or computed, which reduces the dimensionality of the phenotype to a single figure, which can be a number or class. And the different systems that are used most commonly is the European system, which is standardized by the Fédération Cynologique Internationale, and other similar systems are in the US, the Orthopedic Foundation for Animals standard, and in Great Britain and Australia, the standards are defined by the British Veterinary Association. Basically, all of these systems make an aggregate score of various substrates. Then another approach to the phenotype is to measure the degree of joint laxity. And in this category, we have two systems that I am aware of. One is a commercial system developed in the University of Pennsylvania called PenHip. And then we have another system which is very similar to the PenHip, but uses a little bit different instruments, and it's called laxity index, and it's not commercialized to the extent of PenHip. In 2018, these two systems were systematically compared, and this study showed quite good correlation between the two assays. Both assays depend on the distance between femoral head center and the acetabular center when the hips are distracted. That means pulled away from the sockets, and the distance is then divided by the radius of the femoral head. This gives a figure which typically ranges from 0.1 to 1, and a small figure means a good fit, and a large figure means a bad fit, and imposes a risk of developing the disease. Then a few words about the risk factors. Some of the suggested risk factors deal with exercise. It is quite logical and known that good muscle condition can prevent the development and ameliorate the signs of poorly fitting joints. And in young animals, it is considered that free exercise is protective, but for example, going in stairs, it is considered risky and should be avoided if possible. Then nutrition is another risk factor, especially the amount of feed. So if the feed intake is limited, also in the long run, after the animal is fully grown, it has a protective effect on the development of this disease. Young puppies are then quite prone to dietary excess calcium, so this should be taken into consideration because excess calcium can delay the endochondral ossification of the developing joint and or the bones, and also inhibit the skeletal maturation or delay it. It also follows from this nutritional discussion that obesity as such is a risk factor for the disease. There are also some studies which indicate or which point out to the hormones, especially relaxin and estrogen, but I'm not sure if these are all conclusive and maybe not easy to avoid, because some of these studies refer also to the hormones from the milk of the mother. So what happens is that we get this kind of vicious circle where the different risk factors promote joint laxity and increased laxity, then promotes wear and tear, kind of deterioration of the joint, leading to increasingly incongruent articular surfaces and shallowness of the acetabulum, and eventually to various degrees of subluxation and also tear of the articular cartilage. Then about the genetics that we know that contribute to hip dysplasia. Various estimates of heritability have been published, and they vary typically from 0.1 to 0.8. And this means that there is a strong genetic component which plays a role in this disease. Interestingly, the heritability estimates that are based on genomic data are a bit different than those based on pedigrees. Nevertheless, studies have been conducted which have then shown that there are several different loci which are associated with this disease. At least 28 autosomes are involved, perhaps more. The problem with these studies is that the associations have been difficult to validate using independent cohorts. This does not necessarily mean that the studies are not done well, but it could also mean that there are major differences in allele frequencies and the genetic background in different populations, and also maybe different degrees of linkage disequilibrium in different populations and different places in the genome. A notable exception in these risk factors is the fibrillin 2 gene, which is one of the few, maybe the only example where we can find evidence from not only the association but also expression data, which shows that the expression levels of the gene associate with the disease. Then I will describe shortly our own studies. We have been doing an association study in German Shepherds, or actually several, and this figure shows our cohort. In total, 769 animals in different hip dysplasia categories, as indicated. The plot here is what we call a multidimensional scaling plot based on the genomic data, and it shows clearly that the population is divided into different strata. We have in German Shepherds, we have two lines, the working line, which is on the right, and the show line, which is on the left, and then we have some individuals which are what we call a mixed line in the middle. And this shows that the genetic background in these lines is quite different, and it's important to control this stratification when doing the studies. This is a summary of our results. We conducted 5 different studies. In the first line, in the first column, we have the number of animals, and the second column indicates the phenotype, and then the chromosome and the locus in the chromosome and the markers, and then the genes within the loci. The first experiment was on osteoarthritis. So 0 means no osteoarthritis, and then 1 to 3 different degrees of osteoarthritis. The second and third one are measuring the fit of the femoral head and the congruence of the joint surfaces. Basically, it is the distance of the center of the femoral head from the dorsal acetabular edge, and this varies from 5 to 14 minus 5 to 14 millimeters, and the smaller the value, then the better the joint. And then we have 2 studies on two sets of data on the hip dysplasia phenotype as defined by the standard. In the fourth line, we are comparing completely healthy animals with all degrees of disease, and in the fifth line, we are comparing mild disease with moderate to severe disease. And the candidate genes are briefly discussed here. The first candidate gene is called NOX, and it is located in the chromosome 1, and associates with, this locus associates with osteoarthritis and also in the categorized hip dysplasia. NOX3 is a member of NADPH oxidases, which catalyze the production of hydrogen peroxide, which in turn can yield other molecules which are damaging to articular cartilage. But on the other hand, NOX3, to my knowledge, has not been specifically detected as expressed in the articular cartilage. So it remains to be seen what is actually the role of this for the disease. The next candidate gene is called NOG, which encodes for a protein called noggin. And this gene is located in a locus in chromosome 9, and the locus associates with incongruence and severe or moderate to severe phenotype of dysplasia. Noggin belongs to the BMP family of, Noggin inhibits the BMP family morphogens, and these morphogens are known to positively regulate the formation of bone and cartilage. Noggin in particular has been shown to be a key player in the development of bones and joints. And we found markers upstream of noggin, which associate with the severe disease. And then we also find a deletion variant that associates with normal hips or with mild disease. And this variant also inhibits the reporter gene expression of in vitro. So when we looked at these markers in chromosome 9, we see we could identify two loci about 6 megabases apart, and we decided to sequence this region spanning about 7 megabases from 48 animals, and this resequencing yielded a lot of sequence data, and from this we could identify 217 SNP variants, of which 28 were statistically associated with the phenotype. And these 28 variants have been marked with a green vertical bar in this figure. So we see two clusters of significant markers, and in the larger cluster between around 31 megabases, there is quite near to the peak this deletion variant sitting right in the right place. And if we look at this variant a little bit closer, we can see in this figure on the top sequence from human, followed by the sequence in the reference genome from the dog. And then we see 3 variants from our own data. And in the middle, we see this locus of the dog, and then on the human aligned. And the deletion is marked with a blue triangle in the dog locus. And in the corresponding place in the human, we see signs of epigenetic marks which are associated with regulation of gene expression, and also we can see transcription factor binding sites. These data are from the ENCODE project. So with this information, we then proceeded to analyze this deletion variant in vitro. And what we did is we cloned it in front of a reporter gene construct, and we can see that the deletion variants, these are the same same numbers or same symbols as in these sequences A, B, and C. So the A sequence is the longest, and the C, B, and C are deletion variants. We can see that the deletion inhibits the expression gene, expression of the reporter gene in vitro when compared with the full-length variant. And we tested this in two different cell lines. On the left, we have human embryonic kidney cells, and on the right, we have a human osteosarcoma cell line. When we then think how this deletion might be related with the disease, we have this kind of hypothesis that we show that the deletion inhibits the expression of the reporter gene. So if this actually regulates the expression of NOGIN, this means that the deletion variant would increase the levels of BMPs, which then would promote the formation of the bone and cartilage and protect the animal against this wear and tear of the joint. And this is what we see, that the deletion variant is associated with healthy or mild phenotype. Then interestingly, when we compared different reference sequences that we could align, so we can see that from various species, the dog is different from the other species. So it's the second last one is the dog sequence, and this is the boxer sequence, and we don't know what this means. We know that there are different variants in the population, but does it perhaps mean that the reference allele is not the original ancient allele? This we cannot yet say. We will need to study this further. Then the last candidate gene is nanos, located in a locus in chromosome 28. And this gene product regulates the activity of metalloproteinase gene. And this gene product of the MP1 MMP can activate the MP2, which in turn can degrade extracellular matrix in osteoarthritis. But we have no other evidence apart from this association that these are related. The nanos is related to the disease. Then I described shortly the validation study. We looked at the literature and tested 52 SNPs that had been previously associated with the disease using a cohort of 1,572 dogs from 10 breeds. it may be worth mentioning that this cohort does not include German Shepherds. And then we did this association analysis both within and across breeds. And what we could show is that, or we could validate 23 of these 52 markers, which are residing in 21 loci in 14 autosomes. And interestingly, when we look at all the genes that are annotated in these loci, these 250 protein encoding candidate genes, they contain an overrepresentation of a reaction pathway called nedulation, which is ubiquitin, like a small protein that is tagged onto target proteins. And the role of nedulation has been implicated quite a lot in tumors, but also in autoimmune and inflammatory diseases. So this is something that should be perhaps studied further after more validation data. We could validate one of these three loci in the German Shepherd study. So this locus in chromosome 1, which is associated with osteoarthritis. So then briefly, the conclusions. So we identified in the German Shepherd study 3 loci, and one in chromosome 1 was associated with osteoarthritis, and the other one in chromosome 9 was interestingly associated with severe disease when categorized by the radiographs. Then we validated 21 of the 21 loci with 23 markers from the previously identified markers, and the candidate genes in these loci encode for proteins that contain an overrepresentation of the nedulation pathway members. And we can only conclude what is known actually before, that the association depends quite heavily on the population at study. And then the contribution of the individual locus is relatively small and varies also by different populations. Then more generally, when we think about these genome-wide association studies, it's important to realize that it's only a measure of association and by no means tells anything about the causality. It may be causal, maybe not. And if it's not causal, it may be that there is a causal variant linked nearby, and there is a lot of work to reveal all these causal variants. And then it's important also to realize the reliability of these studies, that you have to really carefully make sure that you control for the confounding factors, and this population stratification is an important issue in this respect. Then a few words about the future. I must confess that I'm rather disappointed. We did a lot of work, but what can we really say about it? I think to really study the biology, we need to have bigger samples. And of course, humans are very different from the genetic perspective compared to dogs. But nevertheless, it's exciting and also depressing to read this new study on the human height, where they could show that millions of individuals are needed to saturate the model and to explain all the genetic variation of the common variants. They also concluded that there are more than 7,000 independent loci which contribute to the height of human beings. And if the hip dysplasia is anything similar, we have a long way to go to get a good grip on the biology. We also need denser marker map. There are more denser chips now available. We used Illumina 173,000 marker chip, and imputation has been also successfully applied, which can also give a little bit denser markers. Then I think it should be also discussed what is the perfect phenotype. I mean, how good is the phenotype? Which of the subtraits are really important, and how should we quantify it? There have been also some studies on the biomarkers. So the phenotype is quite complex, and we should consider carefully different ways to reduce the dimensionality of the phenotype to get more power in the analysis. But of course, the reduction in dimensions should not invalidate the measurement. I mean, the phenotype should still represent the disease. And then last, I have listed the references, selected references in my presentation. And then I would like to acknowledge Lea Mikkola, whose PhD work is the basis for this presentation. And also Hannes Lohi, who is a very successful canine genetist in Finland and the Finnish Kennel Club and its member associations. And the financial support from the Academy of Finland and the Canine Health Foundation. Thank you.
Hi, everyone, and thank you so much, Dr. Iivanainen, for that in-depth review of hip dysplasia. Just a couple of points for our audience before I switch over. I want to note that the closing remarks, the agenda has been updated, so closing for the summit will be at 4:45. But we do have time now to take some of the questions that you've asked throughout the presentation and do a Q&A session, at least a brief one, before we can send you off to the closing. So don't forget that you can use the Q&A bar in your seminar window to send those questions through to us. And my apologies, once again, thank you, Dr. Iivanainen, for your presentation and for joining us here at the summit.
Yeah, thank you. So
we've got a, oh, go ahead.
So you are telling me the questions, or yeah?
Yes, I will share with you some of the questions we have already. One of them is around what recommendations do you have for breeders or people that are raising neonate puppies to reduce the chances of environmentally developed hip dysplasia, given some of the studies that you talked about earlier?
Yeah, we haven't studied this ourselves, but what I have understood from the literature, it seems that the young animals' tissues are quite soft, so it would make sense to try to make sure that there are no accidents or bad strains which can cause damage to the soft tissues. And once you have damaged, then it may lead to like a deterioration of the condition because you are making the joint unstable. And of course, one should discourage the calorie intake so that the puppies don't grow too fast, because when they gain weight rapidly, the musculoskeletal system doesn't necessarily keep up with the increased weight. So I think there are studies on the calcium, so you should avoid too much calcium in the feed. That's what comes to my mind.
Yeah, that's actually helpful. I know as a breeder, there's a lot of discussions about how cute puppies are when they're a little chubby and fat. But to your point, we might not be doing the right thing by having them grow that way in terms of preserving the best opportunity for their hips. So keep them lean, it sounds like.
Yeah.
Another question is based on the research, and this gets a little technical, so it might be a misunderstanding, but is it true that you found mild and normal hips sort of clustered together, which might indicate that the difference in those ratings was more environmental versus the genetics signature that you seem to find more correlated with moderate and severe dysplasia?
That's a very good question. I don't want to make that conclusion, but I think our studies indicate that it's possible that there is the genetic risk factors are sort of many and different kinds, and some of them are promoting the early development of hip dysplasia, and you may need to have other risk factors to develop the moderate to severe cases on top of that. And at least in our population, what we studied is noggin variants, or the regulatory variants of the noggin gene. They seem to associate with the moderate to severe forms of the dysplasia.
Right. Okay. Yeah, that'll be interesting to see if there's additional research in that vein.
Yeah.
Have
and
you?
If I may continue a little bit on that, I think it's quite clear that the genetic risk factors are quite many, and they are varying in different populations. So, I mean, depending on the population and the genetic background, some of the risk factors are sort of important for that particular population and not in others. And as a general sort of picture, we know that this disease is polygenic in nature. So it's an oversimplification to look at just one risk factor, genetic risk factor. You have to see the whole picture, and each individual contribution of each different risk factor is varying between populations, and on the general it might be quite small on the overall risk.
Right. Pivoting a little, the next question is there anything in the research or that you could recommend about how to prevent dogs from declining or having degenerative issues with hip dysplasia once they've developed it? Or is it pretty much once it's been diagnosed, we know it's going to get worse? Is there a way to prevent some of the decline, I guess, is the question.
Yeah, I think it's the current understanding that it's a chronic disease. You cannot get rid of it. But of course, if you try to avoid overweight and not, I mean, yeah, I think overweight is maybe the most important thing, that you try to keep the dog lean and in good muscular condition, because if you have strong muscles, you can support the hip even if it's not in the perfect possible shape.
That makes sense.
But I don't think you can get rid of it. Of course, if you are, let's say that the condition is varying, that you're managing to reduce weight if the dog is overweight, I think it can alleviate the symptoms, but it doesn't change the joint.
Right.
Okay. And then it's interesting if you think of a young dog which is still growing. I'm not aware of any studies on that. But if you think about, for example, the elbow joint, young dogs may have sort of a temporary problem with the elbow joints, and once the growth is complete, then the joint sort of aligns properly. So in the young animal, of course, you are in a more dynamic process. So it may be possible that in the future, maybe you can develop some kind of measures to sort of to accentuate the healing process in the growing animal. But I'm not aware of any studies at the present.
Well, we look forward to learning more about hip dysplasia as you and others continue the research. Once again, I'd like to thank you, Dr. Iivanainen, for sharing your research and joining us here at the summit. It's been fascinating. I am a bit sad to say we're out of time here, and this is the last session of the day. But please, for our attendees, do feel free to head over back to the auditorium, and the closing remarks will start in a couple of minutes. And there will be information there on how you can go back to review all of the sessions you have seen or view some of the ones that you weren't able to get to. So once again, thank you, Dr. Iivanainen, and I hope everyone has a great rest of their day.
Thank you.